Whatever the drugs you are taking from external source, all are considered as xenobiotics. Whatever the even foods are also considered as xenobiotics. So you have to go for metabolic process. Then only it will be excreted or eliminated throughout the eliminated from the body through urine or any excretory systems. Okay. So if it will stay long period of time inside the body, whatever, maybe drug or your food particles. So it should be digested. Like food particles should be digested, like your drug, it should be metabolized, then eliminated. So there are your pharmacokinetic study like absorption, distribution, metabolism, and excretion. So all process should be continued one by one. Then only the drug will be active. You can say the drug will have pharmacological actions. Otherwise, it will be a toxic material. So any toxic material is not good for health. So you have to avoid those type of things. Okay. So our aim is to eliminate the drug after administration of the drugs. So the drug should be absorbed, should be distributed and metabolized to produce inactive substances and eliminated through urine or any other systems. Okay. So same thing you have to confirm. So first of all, the drugs are all are lipophilic property. Then it will be converted to hydrophilic drugs. These which are water soluble. First, it should have lipophilic property because it can cross the cell membrane. Okay. So the drug should be lipophilic. After metabolism, it will be converted to hydrophilic, or your hydrophilic means it should be water soluble. So there are two phases involved in your metabolic process, like phase 1 and phase 2. So on phase 1 you have oxidation, reduction, like that. General chemical reactions are taking place. But in phase 2 you have conjugation reactions, glucuronide conjugation, sulfate conjugation, acetylations. Okay. So here it is mentioned that drug metabolite with modified activity. So first of all, the drug will give suitable activity. Suppose anti-epileptic drug or anti-tubercular drug, the drug should have some particular activity. After metabolism, it will give some metabolic products that are maybe biological active or maybe having different biological activity or maybe inactive. So it may be inactive drug, or it will be having some modified activity, or it may have some less activity. So after that, they may be going to phase 2, or directly they may be metabolized by phase 1. Sometimes some drugs are undergoing metabolism through phase 1 followed by phase 2. Some are directly metabolized by phase 1, like that. So examples we can discuss on next slides. So biotransformation is a enzymatic conversion of drug to a new chemical species. The original drug will be converted to some other new drugs or new metabolites, which is having different chemical structure. Okay, suppose aspirin, I will tell you the example of aspirin. It will be converted to salicylic acid in your phase 1. So the chemical structure is different here. Okay, so there will be enzymatic conversion or any chemical modification will be there. So it is a conversion, enzymatic conversion from one form to another form, or one chemical structure to another chemical structure. The new drug product is nothing but your metabolites. is usually a less active compound that is more easily eliminated from the body. So whatever the metabolites are produced by metabolic process or by transformation, that molecule will be less active or less biological active, or may be having different activity also sometimes. Okay. So many things are given under this. So drug metabolism is otherwise also called as detoxification, because if the drug is staying long period of time inside your body system, it will cause your toxic property. It will generate or exert some toxic chemicals. So you have to remove these drug materials by this process, by biotransformation process. That is otherwise called as detoxification. It is nothing but your removal of toxic substance. If the drug is staying for long time, it will cause toxicity. So that you have to remove. Okay. So it is the biochemical changes of the drug and other foreign substances. Your foreign substances may be drug or any other chemicals. Okay. This is leading to the formation of different metabolites with different effect. So due to this metabolic process, it will give different metabolites with different effect. They are not having same pharmacological action. Pharmacological activity may be changed. Okay. Some of the compounds are excreted partially unchanged. In some of the drugs, they are partially unchanged. In similar structure or molecule will be there, and some are known to converted to the products. In some are converted to some metabolic products, and some are also converted to toxic than parent compounds. Okay. As compared to parent compounds, there may be structural changes of the metabolites or metabolic products. So liver is the major site of drug metabolism. So total metabolic process is carried out through your liver. So liver is main the site of detoxification. So all types of toxic materials are metabolized through your liver. So it is the major organ. So, but specific drugs may undergo biotransformation in other tissue. This other part also there may be metabolism, but that is very less. In your small intestine or GIT tract, sometimes metabolism takes place. That is called fast pass metabolism. Means after your oral administration, it is not before going to your liver, it will be metabolized partially by the small intestine. That is called fast pass effect or fast pass metabolism. We will discuss later. So drug metabolism is the biochemical changes occur on drugs or other foreign compounds. Foreign compounds may be your foods or some other materials which are taken from external source. The purpose is which to facilitate the elimination from the body. So the main purpose of drug metabolism is to facilitate the elimination of that substance or foreign materials, maybe your drug or food from the body. So elimination is the main criteria. So you have to eliminate from the body. Otherwise it will be toxic for the body system. Suppose your drug is lipophilic here, you can go for enzymatic reaction, you can go for any chemical reactions like hydrolysis, oxidation, reduction, any type of reactions may be taking place during your metabolic process. So inside the body it will take place, so it will be converted to metabolite, which is in polar nature. First it is non-polar or lipophilic. After metabolism, it will be converted to polar drugs. Okay. After conversion of enzymatic conversion, it will be converted to metabolite of polar nature, and finally excreted. So this is the steps involved in your metabolic process. So without it, xenobiotics can remain indefinitely in the body. Without it means without metabolism, xenobiotics, whatever the drugs or any other food materials are taken from external source, are called as xenobiotics. These are remain indefinitely. So for long period of time, they may stay inside the body. So you have to remove from the body through elimination or excretion process. How they will be excreted? By metabolism only. Without metabolism, they cannot be excreted. So metabolic process is required. It may lead to formation of inactive and non-active drugs. Hence the some network error is there. Okay, so sometimes it may cause your toxic. Just a second, some error is there. Now it is visible because it is visible. Visible now?
Yes, sir.
Okay. So like this, you have to design the molecule properly so that it will not cause any toxic metabolites or any toxic substances. So some of the drugs like haloperidol, it is giving some toxic substances. So it is not good for your health conditions. So you have to maintain or you have to design the molecule properly. Okay. So it should not stay for long period of time. Immediately after giving the action or any biological activity, it should be released from the body. Okay. So why you are going for studying of metabolic process? Why drug metabolism is required? So you have to understand the pharmacological actions and toxicological property of the drug. So if you go for metabolic study, you can understand the pharmacological actions, whatever the activity of the drug, and any toxic property or toxic activity of the drug can be identified. And the importance of shortening the drug's duration of action. So the duration of action should be short period of time. It is good. If it is long period of time, it will cause some toxicity. So you have to go for study of drug duration of actions. It should be shortened. This it should be minimized to short duration of actions so that it will give better activity. Okay, quick onset of action and short duration of action, it is better for the drug. It should not be a long period of time. It will cause toxicity. It will, if it will stay for long period of time, it will cause toxicity. Then the complication of drug-drug interactions. Suppose some patients are taking more than one drugs. So there may be drug-drug interaction. Metabolism is again difficult. So for that case also you have to study the metabolic effects. Okay. It mainly depends on the induction or inhibition of metabolic enzymes. Because due to two or three drugs, there may be inhibition of the enzyme or induction of enzymes. Means they can stimulate or inhibit the enzymes responsible for metabolism. So some of the drugs are may be inhibiting or may be stimulating the enzymes involved in metabolic process. So all these things are required to study during your metabolic process. Okay, metabolism is also responsible for in vivo formation of biological active molecules. In vivo means whatever the activity you are studying inside the body system. In vivo, that is called in vivo. In vitro means outside the body, like your bacterial strain, fungal strain. So in that case, it is not required. Metabolic study is not required. But in vivo study, it is required to go for metabolic study. Means that is most required. Because inside the body system, what is going on, you can find out during the metabolic process. Any pharmacological effect of prodrugs. Prodrugs, again, you have to know about the prodrugs. Prodrugs means they are biologically inactive. After metabolism, they are converted to original drugs. You can remember here. So it is also important for your biological study or metabolic study. So to find out the pharmacological effect of prodrugs, you have to study the metabolic process. Okay, because prodrugs are biologically inactive. After metabolism, they are converted to biologically active drugs. So this is also most essential. Then toxic effects. Any drugs having toxic effect can be identified by your metabolic process or by metabolic studies. Okay. So other things like to find out easily the elimination of the drug, whether the drug is eliminated properly or not. So by biotransformation also you have to find out easily the elimination of drug substances. So elimination is also most required. For that you have to go for biotransformation. Without biotransformation there is no elimination. Okay, directly elimination is not possible. So absorption, distribution, metabolism, then excretion or elimination. Okay, to terminate drug action by inactivating it. So how to terminate the action? Suppose the drug is giving long duration of action followed by toxicity. So you have to terminate the drug action by inactivating it. Means you have to inactivate that drug action by some enzymatic process that is called biotransformation. Okay, because the drug will produce long duration of action, it is not good for health sometimes. It will cause to toxic property. So by changing its physicochemical properties, you can modify or you can terminate the drug action by changing its physicochemical properties by different methods. Means some active drug can be converted to inactive drug, or inactive drug will be converted to active form. So inactive means you can give the example of prodrugs. These are inactive. They can be converted to active form, that is original drug. And sometimes active drug after metabolism converted to inactive. Again same, lipophilic drugs can be converted to hydrophilic drugs after metabolism. Unionized drug, any unionized drug can be converted to ionized drug. Non-polar converted to polar drugs. Some are plasma protein binding. Some drugs they will be converted to free drug. After free only they can be easily eliminated. So these are the general things we have to remember. During biotransformation, these things are mainly required to convert from this stage to this stage. Plasma protein binding form to free form. If they are binding with plasma protein, they cannot be easily eliminated. You have to go for metabolism. So after metabolism, they will be free. Okay. So these things are necessary during your biotransformation process. Okay. Next we will go for some consequence, means how the process or what are the drugs mainly involved during your biotransformation, maybe active to inactive or inactive to active form or equally active form. There is no change in activity. So what are the examples of the drugs available during your biotransformation? Suppose some example is given, active form to inactive form. So you have example like phenobarbital, okay? So your anticonvulsant drugs like phenobarbital, epileptic drugs or sedative hypnotics you have. So phenobarbital, so it will be converted to hydroxyphenobarbital, means hydroxylation process is there. Okay, so it is inactive form. First one is active form, then converted to inactive form. This is one of the examples from active to inactive. Then inactive to active, I told you, inactive means that is a pro-drug always. Okay, that will be converted to active form means original drug. Examples you may get L-dopa to dopamine. So L-dopa is the pro-drug, which is inactive form. Okay, you remember for your exam, GPAT exam. So inactive form of dopamine is L-dopa, which is a pro-drug. Next is parathion. Parathion is converted to para-oxone. Okay. Next, ampicillin is converted to ampicillin. So these are active form of the drug. These are inactive or pro-drugs. So you remember the examples. What are the examples available for this? Okay. Next, active to equally active means there is no change in the activity. Same, they are having same biological actions. Okay. Structure may be changed, but activity will be same. You can see the example of diazepam to oxazepam. Here some changes in the chemical structure, but activity is same. Amitriptyline, nortriptyline, imipramine, desimipramine, codeine, muptin. So these are the examples you remember. Which of the following drugs are equally active after metabolic process? So you can get this example: diazepam to oxazepam, amitriptyline to nortriptyline, imipramine to desimipramine, codeine to muptin. Only slight change in the their chemical structures. Okay, so those things you have to remember. Next, what are the different sites of biotransformation or metabolism? I told you already that liver is the main site for biotransformation or metabolic process. But instead of that, some of the body parts like your small intestine, large intestine, lungs, skin, kidney, nasal mucosa, and brain, they are also having some biotransformation process. So liver is the main clearing house, that is called metabolic process, mainly occurred in the liver for both endogenous and exogenous. Exogenous means xenobiotics, which are obtained from the external source. Endogenous means inside those chemicals are secreted like hormones, okay, or any enzymes. These are mainly metabolized by the liver. But some of the cases like kidney, your intestines are responsible for metabolism. Intestine is considered as initial site of drug metabolism. Initial means in primary stage they are metabolized. Some of the drugs are metabolized in intestine, but mainly in liver. That is called metabolite clearing house. But here it is called initial site of drug metabolism. Means sometimes that is called fast pass metabolism. Means before reaching to the liver, they are metabolized in intestine. That's why it is called initial site of drug metabolism. So that is the things you have to remember. So these are the sites of biotransformation. I told you already fast pass metabolism. So fast pass metabolism, otherwise called as pre-systemic metabolism or fast pass effect. It is otherwise called fast pass effect. Okay, pre-systemic metabolism, pre-systemic. This is the systemic circulation, means blood circulation. So after metabolism in liver, they are coming to the blood circulation. So before reaching to the liver, when you are administering the drug through oral routes, so some of the drugs are metabolized in your small intestine. It is written. After oral administration, many drugs are absorbed from the small intestine, transported via portal system. Hepatic portal system is there. This is portal system to the liver, where they undergo metabolism before reaching to the systemic circulation. So before reaching to circulation, they are metabolized. These drugs, some of the drugs are metabolized before reaching to the systemic circulation. That's why it is called fast pass effect or pre-systemic effect or pre-systemic metabolism. Okay, so you remember these sentences or points for your exam. What is pre-systemic metabolism or fast pass effects? So before reaching to the systemic circulation, they are metabolized in the small intestine. So that is the fast pass effect. So some of the drugs which are undergoing fast pass effect, you remember. Suppose in liver, before reaching to blood circulation, they are metabolized in liver, like isosorbitol, glyceryl trinitide, porphyrin, pethidine, xylocaine, imipramine, amitriptyline, and propranolol. That is a beta blocker. Okay. And in intestinal mucosa like L-dopa, alpha-methyl-dopa, testosterone, progesterone, chlorpromazine, clonazepam, midazolam, cyclosporine. These are the drugs which are metabolized in intestine. And some of the drugs in bronchial mucosa like nicotine and isoprenaline. So these are two drugs available for metabolism in bronchial mucosa. So you should remember the drug names which are undergoing fast pass effects. Okay, so you remember these drugs. So these are the drugs which are having fast pass effects. Next we'll go for so what are the different factors which cause or which affect the biotransformation of the drugs. So you have to consider these are the main factors which are responsible for affecting the biotransformation of the drugs. So first one is your physico-chemical property of the drug. So sometimes some physical property and chemical properties are responsible, whether they are soluble in water or soluble in lipid layers, any melting point or boiling point of the drugs, everything they are considered during metabolism. Any chemical factors like induction of drug metabolizing enzymes, inhibition of drug metabolizing enzymes means some drugs induce the metabolic process or some drugs inhibit the metabolic process by acting on the enzymes. So they may stimulate the enzymatic activity or they may inhibit the enzymatic activity. In that case, you have to consider. Then some environmental chemicals, okay, other environmental factors are there, they will cause the or affect the biotransformation process. Some biological factors like species difference. Suppose some rats are there, Worcester rat, albino rats. So species difference is there. In that case also there is some difference in the metabolic process. Some strains are also different. If strains are different, you have different metabolism. Sex difference, age, diet, there may be alteration in physiological factors like during pregnancy. There will be changes in the body system or hormonal systems. Hormonal imbalance or any disease state, there will be change in the metabolic process. Some temporal factors like circadian rhythm or circanal rhythms. In that case also there will be change in the biotransmission. So these are different factors will be considered during your metabolic process of various drugs. Okay, we will go for some factors. Suppose some genetically also they are altered. Suppose the acetylation of isoniazide and hydrolysis of succinylcholine are genetically controlled. Means by genetic way they are controlled. The hydrolysis process of succinylcholine and acetylation of isoniazide. So they are genetically altered or controlled during the metabolic process. Suppose you will go for chemical properties of the drugs. Some drugs may stimulate or inhibit the metabolism of other drugs. Example given, phenobarbital stimulates the metabolism of diphenyl hydantoin. Okay, so here this drug is stimulating or inducing the metabolic process of diphenyl hydantoin. So this is the chemical property of the drug, how it is affecting the metabolisms. Then if you go for route of administrations, through different routes, suppose you are giving oral route or you are direct intravenous route or parenteral route, how they are affecting the metabolic process. The oral route, for example, can result extensive hepatic metabolism of some drugs. If the drug has high fast metabolism, means if the drug is going for fast metabolism, oral route is mainly responsible. Okay. Suppose in case of diet also, starvation can deplete glycine conjugation. If you are in hungry stage, starvation is there, you are not getting food properly. So it will deplete the glycine conjugation. Glycine conjugation is inhibited. There is less glycine conjugation. Glycine conjugation will be depleted or reduced. Okay, this is a conjugated process or conjugation reaction in phase 2. Glycine conjugation. In that case, it is depleted during starvation if you are not getting suitable foods or nutritions. Okay. Dosage forms. Toxic dose can deplete enzymes needed for detoxification reaction. If your dose is not a therapeutic dose means you are taking the regular dose. If it is a toxic, more dose is there. Suppose your regular dose is exceeded, then it is a toxic dose. So it can deplete the enzymes. Okay, it can deplete the enzymes. So the enzyme action is depleted, means reduced. Okay, so for that detoxification is required. Then suppose age, the liver cannot detoxify chloramphenicol as well as in neonates as it can in adults. So in case of child stage neonates, it cannot detoxify, but in case of adult it can detoxify. Means chloramphenicol can be easily metabolized in liver in adult, not in your neonates. Means in child stage it's not possible. So like this you can consider some of the factors responsible for your biotransformation. Suppose gender, maybe male or female. Young males are more prone to sedation from barbiturate than female. So in case of young males, they are more prone to sedation, means they are feeling sedation by your barbiturates as compared to females. So basically male persons are more sedative than females by using some barbiturates. Some disease state also they are affecting the metabolic process, like liver disease. Suppose liver disease is there, totally liver is not working properly, there will be difficulty in metabolic process automatically. Suppose kidney disease is there, there is decrease in excretion of drug. If there is no excretion, whatever the drug is metabolized also, it will cause toxicity inside the body. It will not excrete properly if kidney is damaged, suppose. So there is both liver and kidney also required for your body system. It should be metabolized by liver properly, followed by your kidney. And kidney should excrete. If kidney is not excreted any drugs, So automatically there will be toxic effect of the drug. The drug is staying all throughout the body, then it will cause some toxic. Then circadian rhythm. Circadian rhythm is in rat and mice, the rate of hepatic metabolism of some drugs followed a diurnal rhythm. This may be true in human as well as so depend on the rhythm also there will be different metabolic process. Metabolism is so it is given example in case of rat mice it is very less, but in case of human being it is fast. That will be studied in your chronopharmacology. That is the study of rhythm in metabolic process. That is called chronopharmacology. Okay. So these are the factors affecting your metabolic process. Okay. Then what are the different phases involved in your drug metabolic process? So already told you there are two phases, phase 1 and phase 2. Under phase 1, you have oxidation, reduction and hydrolysis. Oxidation, reduction and hydrolysis. So there may be addition or uncovering of reactive group. So any reactive group or functional group is there, they may be added or removed. Okay, that is called uncovering. So this process makes the molecule more susceptible towards phase 2. Means after phase 1, they can enter into the phase 2. Okay. So after oxidation or reduction or hydrolysis, they can go for conjugation process. That is your phase 2. Phase 2 is conjugation of endogenous molecule with drug. Endogenous molecules, these are the endogenous substances. They are present inside the body system. They will conjugate with the drug and finally excreted. Okay. So under phase 2, you have glucuronide conjugation, sulfate conjugation, amino acid conjugation, or acetylation or methylation. So any of these process can take place in phase 2 with the drug substances. And that makes the molecule more polar. First off, they are non-polar, converted to more polar drug, and they are finally excreted through kidney. So this is the total process, phase 1 and phase 2. So we have discussed what is metabolism, what are the different factors of metabolism. Okay. So next different phases of metabolism. How the process is going on, you see phase 1 and phase 2. Sometimes it can enter from phase 1 to phase 2, or it can directly enter to the phase 2 also sometimes. Okay. So this is the drug entered through phase 1, undergo oxidation, reduction or hydrolysis. Okay. So following phase 1, the drug may be activated, unchanged or most often inactivated. So in phase 1, they may be activated or unchanged also, or sometimes inactivated. Again enter to the phase 2. Phase 2, they are undergoing conjugation process. Okay, and then the conjugated products are usually inactive form and finally excreted. Here it tells some of the drugs directly enter to the phase 2 also. So here without entering phase 1, they can go for conjugation directly. After conjugation, they will be eliminated through your kidney. Okay. So some are going through phase 1 followed by phase 2. Some are going directly to phase 2. So this is the process. So what are the different enzymes involved in phase 1 biotransformation reactions? So this may be cytochrome P450. This is the most commonly used enzyme. You can go get the question. Which of the following are most commonly used enzymes in metabolic process? Cytochrome or CYP450. You may get CYP450 or cytochrome P450. Another is aldehyde and alcohol dehydrogenase. If any functional group is there, you can use this type of enzymes. Deaminase, amino group is there, you can remove by deaminase. Esterase, amidase, epoxide hydratase. So these are some of the enzymes acting on specific functional groups. Next, enzymes catalyzing the phase 2 biotransformation. What are the different enzymes involved in your phase 2? One is glucuronide transferase, that is required for your glucuronide conjugation. Sulfotransferase, sulfate conjugation, required for sulfate conjugation. So transacylase, that is required for your amino acid conjugation. For acetylation, you have acetylase, ethylase, methylase, glutathione transferase. So you have glutathione here. GSH is given. GSH. This is glutathione. Okay. So for that you have glutathione transferase. So you have to remember which enzyme is responsible for sulfate conjugation. That is sulfotransferase. For glucuronide conjugation, you have glucuronyl transferase. Amino acid conjugation, transacylase. Like that you remember. Then any examples also you can give. Then phases of drug metabolism. So under phase 1, what are the process going on? Under phase 2, what are the process going on? So you have degradative reaction. Degradation will be there. But in phase 2, you have synthetic reactions. So there will be introduction of functional groups like OH, NH2, SH, O, COH. But here you may have conjugation. So conjugates of phase 1 metabolite with glucuronic acid, sulfate, acetyl, methyl group. So with these groups, there may be conjugation with these metabolites because under phase 1 there will be change to phase 2. First the drug will enter to phase 1, then phase 2. So phase 1 metabolite will be conjugated with glucuronic acid, sulfate, acetyl group or methyl group. Okay. And this phase 1 are mainly microsomal, but here microsomal, mitochondrial and cytoplasmic. Inside this system, phase 2 is going on. Okay. Here metabolites formed may be smaller, polar or non-polar, or active or inactive. But in this phase 2, it is metabolites are usually larger, polar, water soluble and inactive. Here inactive or active also sometimes. Here totally inactive. Okay. Because after inactivation only it will be excreted. Next, what are the enzymes, main enzymes in under your phase 1 and what are the reactions? Suppose phase 1 oxygenase. For oxygenase, there will be CYP450, FMO and epoxide hydrolase. So they are responsible for different reactions. Carbon and oxygen oxidation or dealkylation, nitrogen sulfur phosphorus oxidation, flavin monoxidase, FMO, flavin monoxidase. This is required for nitrogen, sulfur and phosphorus oxidation. Epoxide hydrolase, that is required for hydrolysis of epoxides. So like these small examples you can remember. What are the reactions? What are the enzymes required? Under phase 2, you have transferase, suppose. So sulfotransferase. So that is for addition of sulfate. UDP glucuronosyl transferase, UGT is short for, that is for addition of glucuronic acid. If there is addition of glucuronic acid or conjugation of glucuronic acid, this enzyme is required. Then glutathione 5 transferase, addition of glutathione. N-acetyl transferase, addition of acetyl group. Methyl transferase, addition of methyl group. So these are enzymes, how they are responsible for different reactions, you remember. Suppose other enzymes like alcohol dehydrogenase. If alcohol dehydrogenase is there, there will be reduction of alcohol. Alcohol dehydrogenase means reduction of aldehydes. Another is NADPH, quinone oxyreductase, that is reduction of quinones. So you remember all these things. Okay, got the point?
Yes, sir.
Any doubt? So under microsomal and non-microsomal, in microsomal you have oxidation, reduction and hydrolysis. Both cases there is same reactions but different enzymes like CYP dependent, CYP independent oxidation reaction. So these are categorized under phase 1. So you can see some of the drugs which are available under different phase 1 reactions under microsomal. Suppose under microsomal cytochrome P450 oxidation, suppose. So aromatic hydroxylation. So how hydroxylation R will be converted to ROH. So in this case, these are the drugs which are converted to hydroxylation. Phenobarbital, phenytoin, propranolol, amphetamine, warfarin, and another is 17-alpha ethinyl estradiol. So all aromatic compounds are converted to OH with OH. That is aromatic hydroxylation. So this is cytochrome P450 dependent. This enzyme is responsible. That is under oxidation. Another is aliphatic hydroxylation. So this is aliphatic ring. Hydroxylation takes place. Aliphatic group with hydroxylation here. Example, you have ibuprofen, digoxin, secobarbital, chlorpropamide. Okay. So these are example of drugs available for aliphatic hydroxylation. Okay. So you remember the examples. Then microsomal CY450 dependent, that is oxidative dealkylation. Oxidation will be there with the dealkylation, removal of methyl group or ethyl group, any alkyl group. So you are given N dealkylation, N. So here you can see alkyl group is attached, N dealkylation. So for that you have example like MFO, barbital, amitriptyline, morphine, caffeine, theophylline. So dealkylation at N position, dealkylation, methyl or alkyl or any propyl group, all will be removed. Dealkylation, removal at N position. O dealkylation at O position, if it is attached, it will be removed. O alkylation, O methylation here. It is called O methylation. Methyl group is removed. Example is phenacetin, codeine, okay, and paranitro-anisole. This is the examples. S dealkylation means S and S which is attached. That is S methylation here. It is removed out. So RS will be there and HCHO. Example is 6-methyl thiopurine. This is the example of drug. Okay. N oxidation. So N part oxidation will be there. N position oxidation will be there. Okay. Example is aniline, chlorpentamine. Secondary. So this is under primary amine. Next secondary amine you have acetaminophen, 2-acetyl aminofluorine. So these are undergoing N oxidation. At N position oxidation will be there. Tertiary amine, example is nicotine, methaqualone. So these are the examples under N oxidation. Okay. Hydrolysis, suppose hydrolysis will be there, so there will be breakage of this bond. Okay. So this is the ether linkage, ROR, ether linkage. So it will be ROH plus R dash OH. Similarly, amide linkage is there. So it will be RNH, amino group, and ROH by addition of water. So hydrolysis will be there by breaking of this bond. Okay, that is hydrolysis. Again, reduction. Suppose amino group is there. Sorry, nitro group is converted to amino group. NO is converted to here amino group like this. Okay, so this is called reduction. Reduction of nitro group to amino group by addition of hydrogen. Similarly, conjugation and sulfate conjugation, glucuronide conjugation, how it is taking place. So we can discuss on next class. So these are some more time required. And on next time we'll discuss. So here some key examples it is given here. So here you have phase 1 oxidation, reduction, dealkylation, already told you, deamination, hydrolysis, how it is going on. They will produce some inactive products. Then going for phase 2 reaction or conjugation. Then they are eliminated. Similarly, I have given the example of aspirin. So here you can see it is a ester form. So there will be hydrolysis. So it will break down. It will produce salicylic acid. Then the salicylic acid undergo conjugation with glucuronic acid. That is called glucuronide conjugation. Then it is eliminated. So this is the one simple example under your phase 1 and phase 2. So first it is hydrolyzed because ester is there. It will produce salicylic acid, which is biologically inactive form. This is biological active form, which is having antiplatelet and inflammatory activity, but it is not having any activity. Then undergoes glucuronide conjugation, then eliminated throughout the body. So these are the some of the discussions about your phase 1 up to phase 1. Then phase 2 I will discuss on next class. Anybody is having any doubt, you ask. So these things we will discuss on next class, and is phase 2. Phase 1 you have cleared all these things. Okay, any doubts? Anybody any doubts, or will stop the class? Okay, read thoroughly. Anytime, any doubt, you can ask me. Okay.